Introduction. Also thanks to the organizers for giving me the opportunity to present yet another method to infer cancer phylogenies. We already have heard a couple of amazing talks throughout the conference. So let me start with a quick introduction, how I think about cancer evolution. So typically we think of evolution as a process with the power to create structures of incredible functionality. At the same time, also tumors are the product of an evolutionary process, and they are the product of selection and mutation spanning multiple decades. So as you can see here in this cartoon model, so let's assume a normal organ growth, a healthy organ, there's a single cell, and whenever a single cell divides, a few mutations will be acquired. This could be on the genetic level, this could be on the epigenetic levels. However, just very few mutations will have fitness consequences. So you see here a lot of different colors in the cartoon model. Some of these mutations might actually happen in the known cancer driver genes. This could be the yellow ones over here. And if you are unlucky enough, then maybe at some point a precursor lesion will form. Another cancer driver gene mutation is acquired, and a primary tumor grows. And it grows further, and at some point metastases are seeded. So in that talk, I will mostly focus on metastases and metastatic seeding patterns. And what I want to do is basically reconstruct an evolutionary tree based on DNA sequencing samples from spatially distinct metastases. So basically, we would get sequencing samples from the brown metastases and the purple metastases, and we will construct an evolutionary tree looking like that for this cartoon model over here. So we would see basically that these kind of mutations, the yellow ones, green ones, and red ones, those are shared between the two metastases. Due to the infinite sites assumption, we would put those mutations on the trunk, and then the private mutations to these metastases, also they already occurred, some of them even in the primary tumor. With bulk sequencing, we may not actually see these mutations which occurred over here, but we only see them in the metastases. At the same time, you also see that for some of these mutations, we don't really know the order how they were acquired. For example, for those ones and for these ones. However, if we would also get a sample from the primary tumor, not only from the two metastases, we may actually be able to resolve that order, because this pink mutation is actually present in a large fraction of cell, and if we get a sample from that part of the primary tumor, then we could infer that the pink mutation was acquired before the purple mutation. So there are a lot of tools to infer cancer phylogenies, and there are some more traditional tools what I have put here on the left-hand side, and all of these tools have been used to infer cancer phylogenies in many papers, and there is nothing wrong with that. Basically then, if you use these tools, then the samples of the cancer would be put on the leaves, and then you reconstruct an evolutionary tree as you used to do it. So here you are seeing a lot of more sophisticated tools to reconstruct cancer phylogenies. So they are based on variant allele frequencies, cancer cellular fractions, and what they do is basically they also give you the tumor composition. Some of those tools also infer then the phylogenies of these different subclones. And this is by no means a complete list, but you can already see here this is a very crowded area right now, and it's a hot topic how you can do that from DNA sequencing data. So when I started to work on that topic, I wasn't really into computational biology. I was mostly working in mathematical biology. Still, I was facing this kind of problem, and I was working on pancreatic cancer. And if you know pancreatic cancer, if you obtain any biopsy samples from there, then typically the purity is extremely low. So it can vary between 10–15% up to maybe 40 and 50% if you're lucky. So all these very sophisticated tools also basically require that you have some rather nice behaving data, that there is not too much noise in the variant allele frequencies. So we had this idea that we come up with a much more simplistic approach to infer cancer phylogenies, but also a more robust one. And to give you some idea how that works, we basically just develop the Bayesian inference model. We assume, again, a perfect and persistent phylogeny. We have a mixed integer linear program, and then we infer an evolutionary tree. Because we know that an evolutionary tree has to exist, we then basically from our solution can go back to the original data and see that in this data of pancreatic cancer, there are a lot of sequencing artifacts. And not surprisingly, there are a lot of false negatives due to the low purity and due to the low sequencing coverage. So basically, we can then predict all these orange dots, which are underpowered false negatives. Then we can predict powered false negatives, which might be due to copy number changes, or there could be even false positives, which are these pink dots over here. So another important point what I also realized during the construction of our tool Treeomics is even if you have a pretty robust tool, still you can have insufficient data. So one of the trees what we presented in the publication was looking like that. So here on the top you have the normal sample from the pancreas, and then you have 3 spatially distinct lung metastases, 5 spatially distinct liver metastases. However, what you can see on the tree is that the confidence values on the inferred branches are actually very low. So with 2 exceptions, so we are very certain about that cluster over here, and we are very certain about that cluster over here. But otherwise in this tree, the confidence values are below 70%. So how sure can you actually be about the conclusions what you draw from such a phylogeny? So for the review, what I'm working on right now, we actually reanalyzed the data, and now we had whole genome sequencing data. And then, of course, it basically doesn't depend which tool you're using because you have like so much signal to reconstruct an evolutionary tree. And as you can see, still the cluster what we had over here, we were very confident about that one. The same one is still in that new tree, and the same one what I told you about here is also still here, but everything else actually changed quite a bit. So we now have a cluster of all 3 lung metastases with a cluster of 2 samples of the primary tumor, but also we have a distinct liver metastasis which first branched off in this evolutionary tree rather in the middle, now actually diverges first. So even if you would have then a phylogenetic tree with very high confidence values, here maybe you can actually draw some biological conclusions. There's still a lot of limitations to phylogenies. And some of the limitations I'm showing you here. So for example, let's look at this cluster of liver metastases over here. So we know from the topology alone, we don't really know where the ancestors of these different metastases were actually seeding. So one hypothesis, for example, could be that the ancestor of liver metastasis 2 and liver metastasis 5, that one could be seeding in the primary tumor, basically maybe a descendant of the primary tumor region 11. At the same time, this subclone could also be already sitting in the liver and then spreads to those 2 metastases. But also, it could actually be in a lymph node metastasis, which we haven't sampled. So you have to be very careful just by looking at the topology of a phylogenetic tree and then inferring basically seeding patterns. Another limitation of phylogenies would be that, as you can see here, liver metastasis 1 diverges first. However, from that you cannot conclude that liver metastasis 1 is also seeded first, because we don't really know where all these mutations on that branch, so 2,151 mutations, where they were actually acquired. So they could have been acquired in the primary tumor and then the cell disseminated to the liver metastasis, or already this cell here disseminated to the liver metastasis and then these 2,000 mutations were acquired. So for the last few minutes, I just want to change gears a little bit. So what can we actually learn from cancer phylogenies? So I'm much more generally interested in cancer evolution and metastatic seeding patterns. So for that, you have to look at the cohort of reconstructed cancer phylogenies. And this is a very recently published paper where we analyzed cancer phylogenies from 19 colorectal cancer patients. There we had samples of the primary tumor of lymphatic metastases and of liver metastases. And the question what we asked was basically, do lymphatic metastases give rise to distant metastases? And this is a question that has already been posed almost 150 years ago by the German pathologist Rudolf Virchow. And basically, we wanted to use cancer phylogenies to answer that question. And if we reconstructed the cancer phylogeny looking like that, so there is a clade of distant metastases and liver metastases on that branch of the phylogenetic tree, then we would conclude there is a common origin of lymphatic and distant metastases. And if the reconstructed cancer phylogeny would look like here, so there is a lymphatic metastasis clusters together with some region of the primary tumor and distant metastasis cluster together with another region of the primary tumor, then we would conclude distinct origin of trees. So what you can also see here, even if we find common origin, so that is still compatible with sequential seeding of metastases, but also with independent seeding of metastases. So it could be that there is a subclone in the primary tumor which gives independently rise to lymphatic metastases and distant metastases, but still we would basically infer such a phylogenetic tree. So we then did the analysis across the whole cohort, and what we found were basically examples looking like that over here. So this is an example for common origin, where you see all our distant metastases clustered together with some of the lymph node metastases. So this kind of tree would be compatible with sequential seeding of metastases, but also with independent seeding of metastases. However, for trees like this one over here, we actually see that the distant metastasis clusters with one region of the primary tumor and lymphatic metastasis clustered with different regions of the primary tumor. So for that tree, we would conclude independent origin and hence independent seeding of metastases. So when we did the analysis across the whole cohort, then we found that of all the colorectal cancer patients we found distinct origin of lymphatic and distant metastases for 65% of the cases and common origin for 35% of the cases. So with that, I already want to conclude my talk. So I hope I could convince you that you can use cancer phylogenies to address very important questions about cancer biology and metastatic spread. I want to thank all my co-authors, especially Jeff Gerald and Martin Nowak at Harvard, and then Chris, Selwyn, Camilla, and Bert, who generated all this amazing data, which enabled us to come up with these new methods. Thank you very much. Well, we have looked into patient survival also already. The problem is so far the cohorts are extremely small. So I think our cohort was now one of the larger ones where we had multi-region sequencing data, and even that cohort only had 19 patients. So we looked at the survival data, and there are some interesting trends, and you might be able to get a significant p-value, but what really tells us that? So I think we really would need to have a cohort with like 100 patients at the same cancer type at the same stage that we could actually make some reasonable estimates from cancer phylogenies.
Do you have like a teaser of what the interesting trends are?
So I mean, there are multiple correlations. So we saw, for example, a correlation that distinct origin seemed to have a worse survival than common origin. But that also kind of correlated then with early and late metastases, and also with basically how early the metastases diverge on the phylogenetic tree. So what actually is the mechanism behind the survival, we don't really know.